6 or 7.
Hi everyone, my name is Melody.
I'm Alex.
And we decided to explore various page ranking approaches to predicting virally implicated diseases. And so why were we interested in virally implicated disease, for example? Well, we learned over time, after exploring a lot of papers, that Epstein-Barr virus and HPV, human papillomavirus, are associated with approximately 30% of virally implicated diseases, cancer specifically. And so, given that we know that these protein pathways can be really complicated and that there's a lot of different interactions happening that can lead to these diseases, we wanted to explore really these interesting pathways and see what is like a viable or virally implicated disease from EBV and HPV. And so, the significance of our project and why we're going down this path is that there are some existing methods for detecting virally implicated diseases. And this includes kind of looking at the symptoms and being able to connect them with patients who are known to have exposure to these viruses. But we wanted to kind of go to a much larger scope, which involves using protein-protein interaction networks. And so we decided to go down the road of Pagerank because it's already been used in many applications for detecting protein neighborhoods and other complexes within PPI plants. So as you can see kind of in this short little, I guess, equation, Pagerank really utilizes both like the degree of, so basically how many nodes are sending edges into itself, and then that kind of can measure its importance in a network. And so we were kind of hypothesizing that the more important a node is in a network of a virus, that it could potentially lead to other diseases and let us explore more important pathways. So our main methods were primarily constructing like a multi-level protein interaction network. So we had some data sets that were relating various human and viral proteins, as well as disease nodes and their comorbidity rates. And so this was specifically for EBV and HPV, but we think that this could apply to other viruses as well. And then our first step was to run Pagerank to assign a rank to all the nodes. So using that kind of equation and approach that we saw, and you're able to modify some of the parameters as well in this approach. And then we would create edge weights based off the Pagerank of the endpoints, and then run various like, topological algorithms, like shortest path algorithms, for example, to similarly rank the diseases.
Okay, so as Melody just described, we assembled our network, and this is what it would look like. So the red nodes are the viral proteins, the orange nodes are the human intermediate proteins, and the blue nodes are the human proteins that then lead to diseases. And this is a filter version of the graph. The actual version is much more dense because this version only includes human protein nodes and or nodes that have a degree greater than or equal to 5. So it's just like a filtered version for the presentation. But then with this graph, we were able to run, as Melody said, shortest path algorithms to find our virally implicated diseases, or so we predict. And so these are like the results that we got from running our average shortest paths. So essentially what we did is we're interested in finding the paths between the viral proteins and the disease nodes. And so we ran a single shortest path algorithm on each viral protein, and then we averaged out the path length to each disease node. And so with EBV, we found that we were able to identify 3 out of 5. Oh, I guess, sorry for broader context. We used a data set and the results of a paper published by Kovacic et al., which we have, which is in our paper, but essentially we compare our results to theirs, looking at the same like rank percentiles to evaluate our findings. And so we were able to find 3 out of 5 with the same disease, virally implicated diseases within their 5 percentile, top 5 percentile. And although with our approach, we were able to find all virally implicated diseases within the top 56 percentile, whereas Kovacic were able to find them within the top 36 percentile. And then in terms of HPV, we were able to find 2 out of the 2 virally implicated diseases within the top 5%. Although Kovacic et al. Were able to find all of the virally implicated diseases within the top 17%, while we were only able to find within the top 34%. And so overall, we had 4 major approaches, but we only showed the average first pass. Here, we looked at the raw PageRank value, the single source pass from a viral source node, the average first pass, and the frequency of a disease node appearing in the first path. And we found that the average first path is probably like the most promising approach because we were able to find more virally implicated diseases. And this approach can generalize to other protein networks because all we need for the data set, in terms of the data set, is the relationships and connections between the proteins themselves. But as we were going throughout this process, we realized that there are like a lot of parameters that could be better optimized. For example, there's like the damping factor in the Pagerank algorithm. There's a way we assign the weights to the edges in our graph, and we realized if we were to take this further, that a machine learning algorithm or a machine learning approach would be probably better suited for determining these parameters, just because we had to make sort of our best intuitive guess for most of the parameters as we were going through this process. And then some of the limitations that came up throughout this process that a lot of other studies that are using protein-protein interaction networks find are the fact that there's not really like a ground truth data set that we can really validate our results with. We had to pretty much trust Gubacci's validation set that they came up with when we were analyzing our data. And so there's no true data set out there that we can compare it to and say, yeah, these are for sure virally implicated diseases. We pretty much just have to trust. We have to trust Gubacci's work in this case. But yeah, that's it for our presentation, and we're open for any questions.
So number one, where did your PPI data come from? Did anybody actually do protein-protein interaction experiments that include both virus and host?
Yes.
So basically,
Can you go
yeah, sorry.
for all the teams when you switch, just have one on the mic and one to this side.
Sure. Yeah, so basically there are various kind of collaborative data sets that have amassed different protein-protein interactions, specifically for EBV and HBV, which is where they got their data set and where we also kind of pulled it from.
And how is that done? How do you do a PPI experiment across virus and human?
So a lot of it is kind of from experimental results. So it's, I guess they're like unweighted interactions. So it's basically just saying like we found a connection between these two proteins.
But you might not know the answer, but my question is experimentally.
Yeah.
Do you just put all of the proteins from the virus and all the proteins from the human, and you do a full immunoprecipitation, pull it down?
So they looked at like comorbidity and like relative risk rates when they were coming up with their data set, I believe.
No. So these are protein-protein interactions or gene-gene interactions? There's comorbidity.
Yeah, sorry.
So basically, I thought that you were looking at the physical binding of pairs of proteins.
That was part of the.
So when I think protein-protein interaction, that's what I thought.
Yeah. So I guess if we go back to like the construction of the network itself, it has various levels that are where the data sets were coming from different places, basically. So we start with like the viruses connected to their own viral proteins, and then like the virus and host proteins. We're not actually 100% sure how they got these connections. I guess looking back, that we wish we had explored that a little bit more in depth to be able to kind of pre-process the network a little bit better. But yeah.
But basically, right now I'm a little confused because it seems like you're using Pagerank to figure out the network, but I don't know what the original age weights were from.
So yeah, there were no original edge weights in the network. It basically just says like there exists an interaction between these proteins. And then
an interaction would be a genetic interaction, physical interaction, co-expression interaction, comorbidity interaction, just any kind of interaction.
Yeah, and so that's why we wanted to use something like Pagerank to be able to create more, I guess, more detailed edge weights between the nodes in the graph.
Okay, and then in terms of validation, you're certainly showing this bimodal distribution, which is awesome. But my question is there any kind of ground truth that you can use as to what proteins are actually important?
Yeah, so kind of as we explained, a little bit of the difficulty in finding ground truth data is that you're relying basically on clinical like comorbidity rates to say that these diseases are actually related. So so that's kind of how we went about validating our data, which is why there's not exactly, so that's why we were saying like it's not exactly clear if you can basically say like this disease is going to be caused or impacted by this virus, but here's our kind of prediction.
Lastly, your diagrams, a recommendation for all of you guys would be to copy paste them from high resolution. If you go back to your network, so here, you could stretch it to fill the whole screen. You probably don't need the axis label. You should probably label some of the proteins and sort of point out key features there, etc. Great, thank you.